It's my great pleasure this morning to introduce Dr. Heather Whitney from the University of Bristol in the UK. And Dr. Whitney is, she's the reader in plant interactions at Bristol, and she's had a glittering career so far, literally because she studies iridescence, which is the way that certain structures affect the way we see colour. And she was actually one of the first person to observe how certain nanostructures affect the colour on the surface of plant tissues. And for that, she was awarded the Society for Experimental Biology's President's Medal in Plant Sciences back in 2012. She's also received the L'Oreal UNESCO UK and Ireland Women in Science Fellowship. And she's currently continuing her research at Bristol University, where I'm sure she's going to tell us about several different things she's interested in. But she's also a member of the management group of the Bristol Centre for Agricultural Innovation. So she's got a unique insight into connecting nanostructures with biology and then applying it in agriculture, which I think will be really interesting for us. So I'm very happy to hand over to Heather and hear what you're going to share with us today.
Thank you, Stuart, for a very shiny introduction. And thank you so much for inviting me to give this talk. It's really nice, really exciting to be able to talk to you about what I've been up to for several years now. This does cover quite a range of research and it's quite a broad range of research. We're going to be going from structural colour to vision all the way through to then, as you mentioned, the agricultural side of things as well. So as Stuart mentioned, I started off quite a while ago now becoming interested in iridescence. Now, so I'm talking to people who are interested in nanoscience. I probably don't need this slide to explain what structural colour is. However, just as a quick overview, it is a colour that is produced by a nanostructure rather than a pigment. So it is the structure that interacts with light and causes interference of light, rather than having some sort of chemical structure that absorbs certain wavelengths and reflects others. Now, this has been studied for quite a while in animals. So Newton, for example, is the classic example of someone who looked at, so Hooke and Newton and that sort of era of people looked at the structures on peacocks and looked at this, the fact that this is due to structures rather than pigments. And there's a lot known about animal iridescence, where it's extremely widespread. So we see it in mating displays of birds. We can see it in butterflies, all sorts of insects. We see it in fish. Huge range of animals produce iridescence. And that's sort of where my interest in plant nanostructures begins. I was looking at the slightly more, not quite nanostructures on flower petals. I started talking to some physicists about the structures on petals and what impact that they might have on plant-pollinator interactions. And the people I was talking to were actually looking at the physics of natural structural colour in butterflies. I'd never really considered it before, but it suddenly struck me that these structural colours are incredibly vivid, incredibly, they can be incredibly well tuned. And I started to wonder why on earth plants don't use structural colours. I was extremely lucky in that I started actually looking. And what I found is that actually plants do have structural colours, and they have actually quite a range of them. So here's some examples. I don't know if I can. So this is a begonia. This is a selaginella. This is a tulip, and that's a microsorum. So what you have here are a range of different ways of producing iridescence, and in the case of sort of leaf structure versus petal structure, a range of different plant organs that produce iridescence, and for different reasons. Now, I'm not going to be talking about the floral iridescence at all today, the story there is getting much better understood by how it is actually developed. It's produced to help enhance pollination and attract pollinators. What I'm going to be focusing on is this bright blue leaf iridescence. So we normally assume that leaves are green, but if you look, there's quite a range of different, particularly tropical plant species that grow in usually dark conditions that have this amazing metallic blue iridescence. Now, this iridescence was first described by Professor David Lee back in the 1970s. And he described several of these species and sort of said, look, there's this iridescence in plants, normally just being described in animals. What on earth is it there for? And that's the starting point where I started my research. Now, if you look into this, you'll actually find that from a phylogenetic point of view, this iridescence is really widespread. You find it in many of the algal groups. So you find it in brown algae and red algae. You find it in the lower plants, such as Selaginella and lycophytes. There are reports of it also being in mosses, but I've never actually seen an iridescent moss. There's a lot of fern species that appear to have this blue iridescence. Then you find it in both monocots and in dicots. So it's something that appears to have evolved multiple times or been retained, though from the way that we're seeing things, we suspect it's evolved multiple times because there's so many different ways of producing this iridescence. I'm going to be focusing today on one particular way of producing iridescence, and that is the one which we primarily found in begonia. So this was, as I said, first of all described by Professor David Lee, of having this brilliant blue iridescence, as you can see here. So this is Begonia pavonina, known as the peacock begonia, and I think you can see why with that amazing metallic blue. And what David Lee described was the fact that if you look into the begonia leaf that produces this bright blue, in the epidermal cell, so this is the leaf surface here, you actually find some really strange structures that look like flattened pancakes, but with multi-layers in them. And David Lee described these and said, well, this looks to me like multi-layer iridescence. So you've got a structure that is layered in such a way that it interferes with how light is transmitted and reflected from this particular structure. So he had suggested this way in which iridescence was being produced in these leaves, and he suggested it might be something to do with the fact that these plants grow in very low light conditions, and maybe it somehow enhanced photosynthesis. And so this is where I sort of started looking at these plants. Now, part of the problem is that these plants are actually very slow growing. And if you think about, well, if they enhance photosynthesis, why are these plants so incredibly slow growing and not slightly more biologically producing more biomass, sort of efficient photosynthesizers? And so to start answering this question, we started to actually look at the structures, what they're doing, why they're there. So first of all, what we did was to try and work out a way of actually measuring what the iridoplasts are doing, both as a way of producing iridescence and whether or not they were involved at all with photosynthesis. So one of the suggestions was that this iridoplast was a form of modified chloroplast. Whether it's still photosynthesized or not was unknown. So first of all, we wanted to make sure that the iridoplast was actually the source of the iridescence. Now, this is slightly confused by the fact that as well as having these iridoplasts, you also have standard chloroplasts within the leaf tissue. So here I've got sort of a sketch of the begonia leaf. You have standard sort of mesophyll cells here with normal looking chloroplasts, and that's what the normal chloroplast looks like. However, on the epidermis here, you actually find these slightly weird iridoplasts that are thought to cause the iridescence. Now, to actually confirm that the iridescence was caused by these unusual epidermal cell structures, we had to work with some physicists to actually build a microscope that was actually linked to a spectrometer, and we could actually then focus in on an iridoplast specifically, and then measure the light being reflected from that iridoplast at lots of different angles. So we could confirm that the colour being reflected was a structural colour, and that it was the iridoplast rather than any other structure in the plant cell causing that structural colour. And what we were able to do was to get a leaf, to focus in on the iridoplast. And as you can see here, once you actually get these leaves under a microscope, the idea that these iridoplasts are actually the source of this metallic blue iridescence becomes fairly obvious, because they are very bright blue. And we could also then do SEM and TEM to actually model the structure of these particular iridoplasts, to see whether the structure showed the right pattern for the actual reflectance that we were then able to measure using spectrometer. So as you can see here, once you're able to focus in on a specific spot on the iridoplast, you can then get a very clear peak in the blue, showing that these iridoplasts do reflect specifically blue wavelengths. So we got to the point where we knew that iridoplasts were the source of the blue coloration. So they were responsible for the blue iridescence. However, we hadn't confirmed whether or not they had anything to do with photosynthesis. So we had some sort of circumstantial evidence that suggested they were. So first of all, if you look at these iridoplasts under a confocal microscopy and you look at the chlorophyll fluorescence, you see that the iridoplasts do actually show chlorophyll autofluorescence. So these things that look like highly modified chloroplasts do actually still contain chlorophyll. Now, we all know that this doesn't automatically mean that they are involved in photosynthesis, because you get modified chloroplasts turning into things like chromoplasts. And basically, chloroplasts can modify in all sorts of different ways. It doesn't necessarily mean that they're still involved with photosynthesis. But interestingly, what we also found is that, as well as showing chlorophyll autofluorescence and having a structure that made it look like they were modified chloroplasts, they also showed behaviours like chloroplasts. So if you put the begonia's leaves into high light, you actually lost the iridescence. The iridoplasts all moved. So here we've got a begonia leaf where we've put sticky tape, some foil on some portions of the leaf to shade it from bright light, here and here, and then one region where there wasn't any foil over the leaf, and where there was bright light, you can't see any of the blue iridescence because the iridoplasts have all moved in the way that chloroplasts move in high light. And in the shaded regions, the iridescence is still visible. We were then lucky enough to be able to collaborate with Professor Tracy Lawson over in Essex, and she has a PAM setup where you can actually then measure specific photosynthesis rates of specific plant cells. So what this meant we could do is we could do an epidermal peel of the begonia and almost have an internalized control to actually measure from the same leaf and from the same portion of the same leaf, just slightly different layers, whether or not photosynthesis was happening in both the iridoplasts and in the chloroplasts, and if there was any difference in that photosynthetic rate. So if the iridoplasts do carry out photosynthesis, we can measure that using PAM. And if they actually do something different in photosynthesis from chloroplasts that are grown in the same leaf under the same conditions, we can also measure that. So in some ways, we're very lucky that we've actually got an internal control within the same leaf of a non-photonic chloroplast as well as a photonic chloroplast. And using Tracy Lawson's setup, we were able then to measure under a range of different conditions the rate of photosynthesis of an iridoplast compared to a standard mesophyll chloroplast and a guard cell chloroplast as well. And what we found is that for some aspects of photosynthesis, the iridoplasts not only photosynthesize, they actually photosynthesize better than the standard mesophyll chloroplasts. So here I'm showing the quantum yield of those particular chloroplasts, and here is the quantum yield of the iridoplast compared to the mesophyll and the guard cell chloroplast. We were still collaborating with some physicists at the time, and what we were able to then to do is say, okay, if the quantum yield of the iridoplast is better than a standard chloroplast, why is that, and is it something to do with the structure? So we were able then to do some TM of iridoplasts and actually model the structure and how it interacts with light. So, and what's really useful is there's obviously been a lot of work done on chloroplasts. We were so able to actually put into that model a lot of assumptions based on the refractive indices of the thylakoid membrane and the lumen, because a lot of research has already been done on that. So we're able to take a lot of very well-known data and apply it to a relatively simple structure compared to some iridescent structures to actually find out what exactly is happening when the chloroplast actually develops in this way. So using all the parameters, such as the refractive indices, the structure that we've found using TM, we were then able to actually say, okay, what exactly is going on here, and why would a deep shade plant have a structure like this? What does it actually mean? And what we found is that the structural color of the iridoplast actually helps capture light. So when you've got very low levels of light, the photonic structure of the chloroplast actually helps something called slow light, which means that more of the light is captured and is captured more efficiently. And particularly in some of the more green, going towards the green wavelengths, that a deep shade plant would actually predominantly have in a very low light deep shade environment. So basically the photonic structure of these iridoplasts very effectively helps scavenge and scrounge the wavelengths that are actually available to it and make the most of that low light environment and actually photosynthesize where other plants would not be able to, and the photonic structure is a part of that adaptation. So this is an example of what you actually find in a forest. And as you can see, this is where you actually find the maximum absorption of light for a photonic chloroplast. So what we found looking at these photonic chloroplasts is that chloroplasts are capable of forming photonic structures that enhance light harvesting, particularly in deep shade conditions. Now, that sounds like an advantage. So why don't we see more blue plants? Why is it that blue plants aren't found everywhere? If it enhances light collection, that sounds like a distinct, useful thing. So one of the things I've already mentioned is that under high light conditions, the iridoplasts basically skedaddle. They move from being flat and being able to actually capture light to then actually going sideways, where they are then at the side of the cell and aren't actually exposed to high light conditions. So that is a standard response of chloroplasts when they're under high light intensity that would actually cause damage. And again, you find that this plasticity is found not just in photonic chloroplasts, but in sun chloroplasts and shade chloroplasts, where plants can actually develop differences in the structure of the chloroplast, depending on the conditions that they're grown in. Now, this raises other ideas. It might be that actually blue plants are a lot more common than we thought. It's just that the conditions under which we see them, any iridoplast or any photonic structure may not be visible because those light conditions where we can see are just too high. It might also be that there are chloroplasts that show a certain aspect of photonic structure, such as very deep shade chloroplasts, but it's not, the reflectance is too low for us to see that. So it might be that actually more plants than we realise can use this photonic aspect, but it's still not quite as ordered such that we've actually noticed it yet. There's also the fact that in high light conditions, it might well be that capturing extra light would be damaging, and also that further on down the photosynthesis pathway, the operating efficiency of the iridoplast is actually lower than the chloroplast. So there does appear to be a trade-off between having a photonic structure that's really good at capturing light and actually then being able to carry out the rest of the photosynthesis pathway with any efficiency. It's fine if you're a shade plant because there's so little light, you want to capture that light, then you can be actually quite slow for the rest of the photosynthetic pathway. But for a sun plant with lots of light, actually having a low operating efficiency would lead to a lot of damage. So photonic chloroplasts enhance one aspect of photosynthesis as far as we can see, the initial light harvesting, but at a significant cost. They are actually less efficient at other aspects, but that's not really a problem, as most of the plants that we found these really clear photonic structures in are deep shade plants. So overall, it might not be the best adaptation, and it also doesn't explain that while begonias have iridescent iridoplasts, lots of other plants use other ways of producing iridescence. They have multi-layered cell walls. There are lots of different ways that plants can produce iridescence. And this photosynthetic argument doesn't really answer why many of the ferns have iridescent blue leaves, because they're all cell wall iridescence, unless iridescence could actually be multifunctional. And this is where I'm going to take a little bit of a sideways step and introduce you to some art. Could art actually have the answer? And we're going back quite a long way here, back to the start of the 20th century, to a chap called Abbot Thayer, who was an American artist. Now, he did a lot of sort of standard landscapes, paintings and all the rest of it. But one other thing he was absolutely obsessed with was nature, and how you can perceive nature and understand it by using the same concept that an artist would. And he wrote a book in 1909 called Concealing Coloration in the Animal Kingdom. And what he suggests in this book is that brilliantly changeable or metallic colours are among the strongest factors in animals' concealment, and go far towards achieving obliteration without countershading. Hence, iridescence is, as I have said, one of the prime factors of disguise, and quantities of animals profit by it. So what he's suggesting here is that iridescence, whether in plants or in animals, could be a really counterintuitive form of camouflage. Now, he did take this maybe a step or two too far. For example, the fantastical peacock tail, he suggested that was actually a form of camouflage rather than any sort of it was a detectable display, but there could also be a form of camouflage. And he painted this particular picture of a peacock to try and illustrate that. But if you actually look at iridescence in animals, you'll actually find that, yes, you have a lot of animals that use iridescence as a form of display, such as the peacock, but you actually find iridescence in lots of non-reproductive life stages, such as larvae and caterpillars. And you also find iridescence common in monomorphic species. These are ones where the male and female look the same, and so it's nothing to do with sort of sexual display and selection. And so this raised this really supported Thayer's suggestion that iridescence might have an advantage that's got nothing to do with sexual selection. But what could that be? And was he right that it was to do with a weird form of camouflage? And because I was looking at iridescence in plants and looking at the fact that, yes, it has advantages, but there seem to be real trade-offs with it. But if it has additional advantages, such as actually being a form of camouflage and actually confusing a potential herbivore's search image, that multifunctionality could have a really be well selected form. So there are ways that camouflage can work. And I'm lucky enough to work in a department with one of the world experts on camouflage, which is Professor Ines Cuther, and he was lucky enough to then explain to me all this animal side of things, of exactly how camouflage works. So there's ways of preventing detection or recognition, and these can include background matching, where you basically try to blend in, masquerade, where you try to look like something you're not, and disruptive coloration, where you're basically got a very strong pattern on you, where you try to prevent being recognized for basically what you are. So we decided to just go back to the basic concept of, could iridescence act as a form of camouflage? And we used a variety of targets, including iridescent beetle shells, both real and artificial prey targets. And we used 3 different visual systems. We used birds, insects, and humans to see how widespread the impacts of iridescence could be on different visual systems. Started off with bees, and we actually first of all looked at what does iridescence do when it comes to actually detecting, finding a target. And this is what we do, is we put these iridescent targets into a bee flight arena. We give the bees rewards. They want to find more fake flowers like this, and then we actually time them to see how long it takes them to find more targets. And what we actually found was that iridescence actually helps the bees find more flowers. It took them less time to find iridescent targets than it did to find non-iridescent targets, which initially sort of stumped our whole hypothesis that iridescence could be any form of camouflage. But then we actually went out into nature and actually had a look at it. Our bee flight arenas were in standard diffuse light conditions. There was no shade. It was absolutely set up, so there was no shade, standard light throughout. This isn't what you find in nature. If you go out to a woodland, what you actually find is there's lots of different light. That light is frequently moving and changing. So we tried to reproduce that in the lab. We actually used a disco ball to then scatter light all over where we were putting the different targets. And this is the exact sort of environment that you would actually find iridescent plants in. They're not found in sort of open spaces. They're found in deep woodland, deep shade, where you would at most get scattered light and sort of little sun specks here and there. So this is a lot more realistic as to where the plants are, the conditions that they're in. And what you actually then find, if you do the same experiments under dappled light, is that iridescent targets might be less difficult to find under constant light, but more difficult to find under dappled light. So under more natural light conditions, iridescence actually becomes a form of camouflage. We took this a step or two further. We then actually decided to test the iridescence actually out in a woodland under natural dappled light rather than relying on disco balls. And we tried with both bird vision and human vision. So what we first of all did was put out iridescent or then replica targets which weren't actually iridescent but showed a range of the colours that the iridescence showed to make sure that it isn't the colour that's the camouflage here, it's actually the iridescence. And for the birds, we then hid a mealworm behind whichever target, so the birds would know to look for this sort of object because there was a reward behind it. For the humans, we gave them a laser pointer and asked them to actually find any of these targets that they could. And in both cases, what you actually find is that both the birds and the humans find the iridescence and the black targets the hardest to find. So the purple target showed maximum mortality. So these are the number of dead mealworms that are actually eaten, so that the prey items that would have been eaten if they were actually prey. Whereas the fewest ones that actually eaten were from the iridescent casing or from the black casing, and the detection probability for both iridescence and black is a lot lower than any of the colours that are, if they're statically produced by the iridescence. So it's not the colour, it is the iridescence that appears to actually produce a rather effective form of camouflage. So it looks like iridescence can be a very counterintuitive way of avoiding being predated, whether you're an insect or potentially an iridescent plant. So Thayer probably did get this idea very right. Iridescence may obstruct prey detection under some light conditions, and it may explain the widespread occurrence of iridescence in many monomorphic animal species and potentially in some iridescent plants. So can we now test this? Can we actually say, okay, so iridescence can enhance photosynthesis and it can act as a form of camouflage in plants as well, as a way of deterring or stopping herbivory? And this is where I shift onto a slightly different range of nanomaterials, because we hit a major brick wall here. One of the things I was hoping to do with looking at iridescent plants was actually start manipulating the genes, and I see if I could either produce iridescent plants to test some of these hypotheses or knock out the iridescence to actually see if it altered the photosynthesis or altered the rate at which they were then herbivores interacted with them. And what I found is that all the iridescent species I tried to grow were completely genetically recalcitrant. Basically, you cannot transform iridescent begonia, however you try it, and we tried it every which way we could for 5 years. You cannot transform these begonia. But at the time, when I was just starting to give up on this whole idea, I had recently started a new collaboration with a colleague over in School of Chemistry, Professor Carmen Gallen, and we were actually starting to look at synthetic nanomaterials. Originally, we were looking at these to see if we could manipulate light in plants in a similar way to the iridoplasts, to see if we could optimize that light manipulation to get a way of helping to collect light, but without then that trade-off that iridoplasts show. So we were looking at different nanomaterials to see if we could manipulate light in plants. This isn't a particularly novel idea, so this is an idea that some amazing papers have been published on, this idea of plant nanobionics. So you can put nanomaterials into plants, and then by making those very specific, you can get a whole range of functions, from augmenting photosynthesis to actually using them as sensors to detect whatever you want to. So it's something that we knew could have potential. We just wanted to try things a different way. Where we started was what was a growing area at the moment, the idea of quantum dots. This idea that you've got highly fluorescent nanomaterials, and that you can then track them, and that those then might be able to interact with the photosynthesis centers in some way, and that the fluorescence might actually be able to enhance photosynthesis. And if you look, you can actually buy a lot of these quantum dots now, and they say that they're long-term, they're photostable for long amounts of time, and they've got this brilliant fluorescence, and it all sounds amazing. So we tried to make quantum dots and put them into plants. making the quantum dots went well. We were able to make fluorescent quantum dots with a cadmium core and with a nice strong photoluminescence. And there's a very established pathway for synthesis and functionalization of these quantum dots. However, there's always been a bit of concern that there might be cadmium leaching and toxicity. And that's what we found. This is a very dead plant. This is a plant that we basically tried to add our quantum dots to, and basically, while our quantum dots showed a couple of hours of photostability, if you actually put them in plants that are then in a 16-hour photo period, they're not photostable for that long, and you end up with basically the cadmium very effectively killing the plant. So what we found is that for what we wanted to do, quantum dots, we couldn't get anywhere with them because we ended up with dead plants. So we started to look at something a bit different, and Carmen's lab had recently developed a new synthesis method for making something called carbon quantum dots. So again, these are very small nanomaterials of 10 nanometers or smaller. They are fluorescent. They don't have quite the same quantum yield as the quantum dots, but they are fluorescent. Because they're basically only made out of carbon, they don't have that cadmium toxicity issue, and they are a lot more photostable compared to the quantum dots. The other advantage we found is that they can be functionalized. So as well as making this core of these fluorescent carbon dots, you can then basically stick on a whole variety of different functionalization molecules around that core carbon dot, and that can give it different properties. So what we did was to start off with both the core carbon dot and then also stick glucose onto the carbon dot and basically pop that into wheat. And it really, as I'll explain again in a few minutes, what is also very clear is that these are taken up incredibly effectively by plants. So you can water these on, you can spray these on, and they are highly water soluble and are taken up very effectively by plants. And to cut a long story short, in the case of these carbon dots, what we found is they were taken up incredibly effectively. And for the glucose carbon dot, they actually enhanced photosynthesis significantly, to the point that when they were applied to wheat, you ended up with around a 20% increase in yield of the wheat grown in the greenhouse. So in some ways, we'd done what we set out to do. We'd found a nanomaterial that did appear to enhance photosynthesis. And here you can see the take-up of the carbon dots showing some of the blue fluorescence. And as you can see, it's taken up through the vascular system of the plant, and then it does go then to throughout the plant. It's transported really effectively. So, but this did then raise another question. Carbon dots are taken up into the plant by multiple routes really effectively. Could they potentially carry something with them? And I've mentioned the fact that my iridescent begonias were completely genetically recalcitrant, and that this meant I couldn't get the gold standard of proof as far as could they be iridescent partly to protect against herbivory. But it's not just my slightly strange model system that suffers from this issue. There's quite a few plant species where plant transformation is this huge it slows it. It's a real knot point in the system, and it just slows everything down. It can take, for example, over a year to transform sorghum, and there are lots of other plant species or varieties that just are genetically recalcitrant, either can't be transformed or really inefficient at being transformed, and could really new ways of plant transformation could be of huge benefit. And there's a really good review on this by Alt-Peter et al. Back in 2016. So we started having a think about this. Again, out in the literature is the idea that, again, nanomaterials could be used to deliver nucleotides to plants. So this an excellent paper had then come out in, it was actually came out in 2016 and properly published in 2017, using clay nanosheets to deliver double-stranded RNA, where you spray on these this nanoclay, it goes onto the leaf and then acts as a reservoir for double-stranded RNA, which can then do impressive things like help protect against plant viruses. So we're seeing that there is a need for thinking about using nanomaterials to enhance plant transformation, and actually it looks like it might be possible. So we decided to test it. And so what we did is we got a fairly standard plasmid. We altered the functionalization of the carbon dots so that they had a peg chain attached. What this meant is that you then had the negative charge, the DNA, the positive charge, the peg, and then an electrostatic interaction between the two. We did some initial work on seeing whether they formed a nanoplex. It appeared that they did. So we then decided just to try it out. So with a plasmid containing a YFP, a vector that it would express a YFP protein, yellow fluorescent protein, we basically got a chunk of Arabidopsis, dunked it in some water that had the plasmid carbon dot nanoplex, let the plant sit in this water for a bit, and then imaged it under a confocal microscope. And what you find is that actually under a confocal, you can see the autofluorescence of the chlorophyll, you can see the blue fluorescence of the carbon dot, and then you can also see the yellow fluorescence of the YFP being expressed. So basically what it's suggesting is that the carbon dots are taken up with their own fluorescence, so you can then track that, and then it is delivering a plasmid with YFP, which is then being expressed, and you can then actually image that. And we're also lucky enough to be in a department with Professor Keith Edwards, who had been working on gene editing in wheat and had a plasmid that had a nuclear targeted GFP and the gene editing machinery. And so we were able then to try this with basically the same thing, where you literally take carbon dot, take the plasmid with the nuclear targeted GFP and the gene editing machinery all in one plasmid. You spray that onto wheat. You then look for the nuclear targeted GFP, suggesting that the plasmid has been successfully delivered and expressed. And then you can then basically have a look for any gene editing. And although the band is very weak, if you then basically extract that you can then do PCR to actually show that the editing has occurred, sequence it, and you find that editing has occurred in some regions of the wheat leaf. Obviously, it's not a universal edit, it only happens in individual cells. So you can end up with a bit of a chameleon type plant. But you do end up with both expression of the nuclear targeted GFP and the machinery for editing in a specific SPO11 gene. And you then see that editing has actually occurred. So by using sort of nanomaterials that we originally designed to try and enhance photosynthesis, we seem to have accidentally developed a plant transformation system using carbon dots, which themselves enhance plant growth, to carry plant expression plasmids into plant cells. And this is, so far we've managed to express both reporter genes and the machinery needed for gene editing, leading sort of spray-on gene editing. And we're hoping to now start to use this technology to continue to investigate the function of leaf iridescence. I've mentioned a lot of people during this talk, and particularly, so this is mainly the people who worked on the leaf iridescence, in particular Martin, who did all the modelling and in collaboration with Ruth, Matt, who did a lot of the work on the initial work on the begonia iridescence, and then Jay then looked at the diversity of it. Nathan looked at this in some of the lower plants, such as the Laginella, and Kara's been working on both the iridescent plants and the carbon dots. All the iridescence camouflage work was done predominantly by Karin Jernsmo and Joe Hall, in collaboration with Innes Cuffill and Nick Scott Samuel, who's a visual psychologist. And we also did a lot of measurements of iridescence using a large robot arm with help from an engineer, Henry Knowles. And then the carbon dot transformation method was developed in collaboration with Carmen Gallen, and particularly with Tom Swift working on the photosynthesize and Katie Higginbottom working on the transformation side of things. And thank you very much for listening.
Thank you, Heather. Thank you for a fascinating talk, giving a real insight into some different areas of life from what we normally look at. I'll open the floor up to questions. Just while everyone else is thinking, I have a quick one, which is, I know that you mentioned trying to improve photosynthesis efficiency in plants by using nanomaterials. Do you think there's the iridoplasts, so the sort of naturally occurring photosynthetic improvers, as it were, do you think those can be moderated? You know, if there are things to do with changing the spacing in the iridoplast or something that can affect the photonic characteristics?
Yes, I do think that they have the potential to be developmentally plastic. And I do think that if this is I want to be able to genetically transform begonia, if we can find the genes that control that, we can actually then see if we can take advantage of that plasticity. It might be that it works better in some light conditions than others. So we do see that there is some, there is some developmental plasticity in it. There is some diversity as to the exact structuring of the iridoplast from different species, from slightly different ecosystems. So there's diversity, plasticity of diversity, both within a single species, but then even more diversity between different species.
Great, thank you. Who else would like to ask something? Okay, I do have one more question, and then we'll let you go. But so I know you showed the carbon dots being used to take DNA into the plants. Have you tried doing the same with RNA yet, like the clay nanoparticles you mentioned?
We haven't, but there has been a paper out using carbon dots for that approach. Yes, so I can't remember. So I can't remember the people who I think Schultz et al. But yes, that has been done. I think they use a slightly different functionalization, but it is possible to take. So basically, nucleotides and carbon dots appear to work. Whether it's I mean, smaller ones should be actually taken up more effectively. There is a size limit, quite a large size limit, but we can get quite large plasmids in, but there is a size limit to the plasmids we can get in at the moment. And it's so small bits of DNA seem to get in more effectively, or RNA get in as well.
Great. Okay. Well, if there are no other last minute questions, I'd just like to say thank you very much on behalf of Sunum for giving this seminar as part of our Nano Open series. It's been really interesting, and we appreciate you taking the time for us.
Thank you very much for inviting me. It's been really nice to be able to talk.
I wish everyone a good day.
Thank you.
Thank you very much.